Hello? Okay. Yeah, I think it's okay. So hello everyone. My name is Olena. I'm a PhD student from Faculty of Sciences, University of Porto, and I'm taking the doctoral program in sustainable chemistry. And today I will talk a little bit about a part of my PhD research, which is to produce control release fertilizers from Vineyard Prunings Biochars. Okay, so why are we interested in biochar? So biochar can be produced from agricultural and animal wastes, also from sewage sludge and from biomass from forests. It is produced in high temperatures and in low oxygen environments, and it has a high surface area, presence of functional groups, and a high absorption capacity, which allows us to absorb nutrients essential for plants. And currently, these materials have no economic value, so this could be also a way of adding some value to them. Why do we want to produce control list fertilizers? So currently, the conventional fertilizers have some problems associated with them, as you know, including leaching, runoff, and evaporation. And in some cases, the farmers apply higher quantities to guarantee that the nutrients are delivered to the plants, but this can create some overdosing and also runoff, which causes eutrophication. In that sense, if we could produce control release fertilizers that deliver them over time, we would be following the principle of economic economy, because we are adding value to a waste that currently has none. And we can also improve the physical properties of soil, since biochar can also have some advantages in that sense. In our case, we used vineyard prunings for this application because in Portugal, especially in Porto, we produce a lot of wine, and therefore we have a high quantity of this residue generating there. For our work, we produced 4 different biochars. So one of them was in a company, so it was produced in larger scale. Another one in a CO2 environment, and the other two we were produced with magnesium chloride and aluminum chloride. Prior to the adsorption, they were added to the biochars, then to the vine canes, then the vine canes were dried and paralyzed. So for the absorption of the nutrients, we in the beginning had some problems. So we ended up performing the studies at different pH values from 2 to 13 to see if the pH influenced the capacity of absorbing the nutrients by the biochars. And what we observed was that indeed it had an influence. In the case of nitrogen, we were able to absorb higher capacities at lower pH values. And for this case, we performed the following studies using the biochar produced at the industrial ovens, because we could produce higher amounts and also had the higher absorption capacity. And in the case of phosphorus, we performed the following studies with a biochar modified with magnesium chloride at pH 8. After determining the best pH, we optimized other parameters, which were the biochar weight to solution ratio, the amount of nutrient absorbed in function of the contact time, and also the initial concentrations of nitrogen and phosphorus in the solution. And the final results that we obtained for the adsorptions could be described both by the Langmuir and the Frederick models, but the Langmuir model was the one that fitted best with the results. And in this case, we were able to determine the maximum adsorption capacity of 10.4 milligrams of nitrogen per gram of biochar and 12.7 milligrams of phosphorus per gram of biochar. Following, we performed the adsorption studies, and in this case, we performed them in a column. The column was used with with soil and 3% of biochar. The column was saturated with water, and then we passed 140 milliliters of water per day. And the studies were the data was collected from 1 to 72 hours. In this case, 3 types of biochar were tested. The 2 ones that were enriched with the nutrients, and the biochar produced by the industrial ovens. To see the difference between the enriched ones and the one that was not. And also we performed a study with only soil to serve as a control. Here are the results. As we can see, the difference between the biochar produced at the industrial ovens that was not enriched and between the soil is not very considerable. So even though the biochar is produced from matrix that supposedly has some nutrients in it, it's not sufficient to deliver the required nutrients to the plants. As for the ones that were enriched, as we can see, both nitrogen and phosphorus released both biochar released nitrogen and phosphorus over time. In this case, after 24 hours, there was no much nutrient being released. And even though this was interesting, because even though phosphorus was the one that was present in higher quantities in the biochar, the release of it was lower than nitrogen. So what we think and propose is that it's more efficiently bound to the biochar, and probably over a longer period of time, it will release even more. So with this experiment, we were able to conclude that vineyard pruning biochars are able to absorb plant nutrients and afterwards release them over time. They can be a more environmentally friendly alternative to the conventional ones. This is also a way of giving some value to these residues that currently have none. And in this case, we tried with nitrogen and phosphorus, but we could also try with other nutrients that are lacking in the soil. And therefore, they can be adapted to the plant and soil necessities. So these are the acknowledgements for the funding of this work. And yeah, thank you for your attention.